Okay. In today's topic, I'll talk about another process by which molecules and ions they get transferred across molecules or ions. They get transferred across the cell membrane against the concentration gradient. So far I have been discussing in diffusion and osmosis. In both of these cases, I discussed in both of these processes I said that the molecules go along their concentration gradient, which is from a region of their higher concentration to a region of their lower concentration along their gradient of concentration. But here in active transport, what happens? Active transport, the molecules are transferred across the membrane against their concentration gradient, which means if these are the molecules over here, and imagine this is the extracellular matrix. Imagine this is the extracellular matrix, and imagine this is the cytosol, which is the inside of the cell, the cytoplasm, the liquid part of the cytoplasm. Okay, cytosol. So these particles, these yellow colored particles are present in this many concentrations outside, and they are present in higher concentration inside. But despite such a negative concentration gradient, the particles will be transported. But here, because you're transporting the particles uphill, you're going to need energy. The particles are being transported against concentration gradient, so uphill. So we are going to need energy for that. That energy can come from various sources. Normally, when we talk about active transport, and when we talk about active transport, we mean the process by which molecules are transferred across or moved across the cell membrane against their concentration gradient using energy from hydrolysis of ATP. ATP is the energy currency of the cell. That is, all our metabolic reactions taking place inside the cells, ATP is the source of energy to drive those processes. Energy currency of the cell. So this ATP is hydrolyzed. Hydrolysis means when you use water to split a molecule, so hydrolysis of that will produce two products. One is referred to as inorganic phosphate and the other is adenosine diphosphate. By the way, those who don't know the full form of ATP, it is adenosine triphosphate. So when we talk about primary form of active transport, because there are other kinds of active transports too, in which case the energy sources to drive the movement of molecules against concentration gradient, it comes from different sources other than ATP. When it is mainly by the hydrolysis of ATP, we refer to that process as primary active transport. Okay, so here you have the membrane, and here you had many molecules, and here you had molecules of the same kind but in much higher concentration, and this movement will be driven by hydrolysis of ATP. Now let us look at an example of that. We'll talk about sodium potassium pump. Pump is the term which is given to the proteins which form channels as they pass through the lipid bilayer. So if this is the lipid bilayer, so these proteins will be present across the membrane, okay, embedded in the lipid bilayer. Imagine this to be the sodium potassium pump. What happens is that this pump this protein channel, it drives the movement of both sodium and potassium against their concentration gradients. But the energy here, it comes from hydrolysis of ATP. So this is ATP and it is hydrolyzed to ADP and inorganic phosphate, and the energy which is generated by the hydrolysis process, it helps in driving the molecules. Let's say sodium, it's present here. Okay, first I'll refer to this side as cytosol to be clear, and this side is extracellular matrix, which means outside the cell. So here the sodium ions will move out, and for every 3 sodium ions, the number of potassium ions that will move in will be 2. So 3 sodium ions move out, for every 3, 2 potassium ions move in. But both of their respective movements are going to take place against concentration gradient, which means this side you'll have fewer sodium and that side you'll have fewer potassium. So the movement, I hope I'm drawing it clearly, and I hope it is depicting what I'm trying to say very well. So here you have sodium in its higher concentration, here it's high, here it's low, and it is moving against the concentration gradient. And here potassium is low and here potassium high, and it is going against concentration gradient. The energy is coming from hydrolysis. This is mainly categorized under primary active transport. Now let's move on to the other kind. So if there is a primary, so you must be guessing that there has to be a secondary, right? So yes, there is a secondary active transport. This secondary active transport, it does not rely on the energy generated from hydrolysis of ATP. ATP molecules are not needed here. So secondary active transport, what is the source of energy in this case? The energy source is co-transported particles. These could be atoms, ions. So I'm referring to them by a general term, particles. Okay, co-transported particle. This co-transported particle is going to be transferred along the concentration gradient. So let me draw a diagram here. So if this is the lipid bilayer, yeah, and if this is the protein through which the molecules are going to pass, in secondary active transport, or these are also referred to as coupled transporters. Yeah, coupled as in the target molecule, whatever the target molecule is, let's say I'm going to use this color to represent the target molecules. So this is the target molecule, and along with that, another molecule will be transferred, which will be the co-transported molecule. The co-transported molecule will move along concentration gradient, along concentration gradient, whereas the target molecule will go against concentration gradient. So this is the purpose here. I am moving one molecule along the concentration gradient, using which the energy which is going to be generated, that is the energy with this other molecule, the red colored molecule, will be using to get transferred, but its transfer will be against concentration gradient. So active transport against concentration gradient, but the energy source is different. Okay, so there are two kinds of such transports. One is referred to as the symport. Okay, I'm going to do one thing. I'm going to draw it here. Hmm. I'm going to draw it here to keep it clear and simple. So first I'm going to, so this is your symport and the other one is antiport. As the name suggests, sym and anti, sym means both the target and the co-transported particle will go in the same direction. Anti means they will go in opposite direction. Now let's look at a diagram to understand it better. Okay, so this is your lipid bilayer, and this is also the lipid bilayer. Now let me draw. Okay, so let's say this is the protein. I'm drawing very simple diagrams to help convey the concept, but you should know in reality the proteins, the channel proteins or the carrier proteins or whatever kind of protein that passes through the lipid bilayer, that is embedded in the lipid bilayer, those proteins are not that simple. These are schematic diagrams, okay? Just so you know. So this is a transporter, particularly known as a co-transporter, and this is an antiporter, also known as an exchanger. Okay, exchanger. And symporters are sometimes also referred to as co-transporters. Okay, now here, so our, okay, so this is our target molecule, and it is going to go against the concentration gradient. Let's imagine this is the inside of the cell and this is the outside of the cell. Okay, so this is also the inside of the cell, outside. And you have our co-transported molecule which is going to go along its concentration gradient. So you have many of those here and very few. Now this molecule is going along its concentration gradient, and it is this molecule's energy which will be used by the target molecule to get it through, to pass the cell membrane, okay, to go through the channel. And in case of antiport, here, this is our, okay, first I'll draw the target. So target is going to go against concentration gradient. You have many, and the target's job is to go inside. It wants to go inside. Now the co-transported will go along its concentration gradient through to give the energy, but it will go in the opposite direction to the target molecule. Okay, so that is what simply symporters, antiporters do. These are coupled transports. This is also active transport, by the way. Coming to the third kind, which is driven by light energy. Okay? And the proteins are called light driven pumps. An example of this would be that we have seen in a bacteria called Halobacterium salinarum. This bacteria, you can also look at the picture, it will give you a better understanding. This bacteria has got purple patches on its cell membrane. From the outside, if you look at this bacteria, you will see that it has got these purple patches. These purple patches are due to the presence of a protein, a transporter basically, which is called bacteriorhodopsin. Now, if you imagine that I have cut this section and I have magnified it over here. So if this is the plasma membrane, cell membrane of the bacteria, and if this is the protein, if this is the channel through which the pumping is going to happen, right? Let me clear this part and this part. Okay, better now? Now, what happens is that it absorbs light energy. So light energy, certain wavelengths from the light energy, sunlight, okay, to drive the pumping of protons, that is H plus ions, okay, against concentration gradient from inside of the cell to the outside. So against concentration gradient would mean that if you have this many protons on the inside, you will have many protons already on the outside. So normally it shouldn't happen, but it is happening. So active transport. But the energy source is not ATP here. The energy source is light. So this is another kind of active transport. Now we move on to the fourth kind, which relies on redox reactions. Redox reactions, those of you who need more clarity on what redox reactions are reactions that simultaneously undergo oxidation and reduction. Oxidation is when an atom, or it could also be an ion, a particle loses electrons. Okay, so it loses electrons. We say that that particular particle has undergone oxidation. It is undergoing oxidation. It has lost electrons to produce a certain ion. Let's say copper atom. Copper atom. Okay, let's say copper atom. It will produce copper ions by losing electrons. Okay, so when it loses electrons, two electrons, it produces copper ion. So it is getting oxidized. And reduction is, we say something is undergoing reduction when it gains electrons, right? When it gains electrons. So let's say if there is an ion, let's say zinc ion is gaining two electrons to give you zinc atom. This is reduction. Now there are reactions in which particles, some particles are undergoing oxidation, whereas other reactants are undergoing reduction. Such reactions are called redox reactions. Now, in living organisms as well, there are many redox reactions which occur. The redox reaction driven pump that I'm talking about, it is referring to, an example of that would be, I'd like to use the example of electron transport chain and oxidative phosphorylation. But I'll talk about it briefly over here so as to not make this topic overwhelming. But basically, if I have to put it simply, uh, I'll talk about this process, by the way, in another video, comprehensive video on this particularly. Okay, sorry about that abrupt cut. My computer was lagging. Anyway, coming back to this. So, this process is something that occurs along the inner membrane. These protein complexes, this electron transport chain that I'm referring to here, it's a chain of complexes, protein complexes, which are present on the inner membrane of mitochondria. Okay, so I'm trying to draw a simple diagram of mitochondria. So here you have a mitochondria. So these proteins are present over here. These complexes are present over here on the inner membrane of mitochondria. Now, these complexes, they have a very important job. What they do is that they undergo redox reactions, and by doing so, they transfer electrons through them from the first one to the last one. Like, that is how they are arranged over there. And the details of how it serves a purpose, I'll talk about again, like I said, in another video. But this video is about active transport uh, using other kinds of energy sources. So this fourth one relies on redox reactions. How? So redox reactions involve transfer of electrons. So all of these complexes, they undergo redox reaction, and electrons get transferred from one end to the other. And eventually the electron combines, the electrons which are transferred, it combines with oxygen and protons to give you water. Electrons combine with oxygen, let me draw it simply, and protons to give us water, which is one of the products of respiration, right? So respiration occurs in mitochondria. So these protein complexes are important in bringing about that process. Okay? So when electrons are transferred, what happens is that it pumps protons, it pumps protons from the matrix. This region is called the matrix of mitochondria. Okay? Here you have mitochondrial DNA, mitochondrial ribosomes, proteins, everything over here. Yeah, these protons are pumped from the matrix into the intermembrane space. Now this creates a proton gradient, and that proton gradient is responsible for again transfer of protons through another protein which is also present in that line of protein complexes. And when it passes through that protein complex, when it passes through that protein, it helps in the synthesis of ATP. Okay, but the details again in another video. Here the important thing over here is that these redox reactions which are occurring over here, that is giving the energy to pump protons against their concentration gradient from the matrix into the intermembrane space. So technically this is also active transport, but here again ATP is not utilized. What is utilized is the energy which is generated from the transfer of electrons across the protein complexes. Okay, so that is all. These are a couple of types of active transports, including the primary. I have also talked about other types, and I hope this video was fun and it was clear. If you have any questions, drop your questions in the comments and I'll try to answer them as much as I can. Until then, thank you for watching and take care.